I'm going to go over the instructions for writing your osmosis lab report. I would, on a separate sheet of paper, I would jot down some notes so that you have an idea of what to expect when writing your osmosis lab report. First and foremost, this is the format of your lab report, so this is what I'm looking for in terms of format. So the title, it has to have a title. A good title is the effect of the independent variable on the dependent variable. You should already know what the independent variable for the osmosis lab is because we went over it before you guys started the lab. You should also know what the dependent variable is because, again, we also went over it before we started the lab. Do not, by any circumstances, put as your title the effect of the IV on the DV. The last lab report that we did, I had some students actually physically write the effect of IV on DV. I don't know why, but that is not an appropriate title. You need to insert the proper independent variable and you need to insert the proper dependent variable for the title. So please don't make that mistake this time around. Next, you should be writing down your question. This should already be done, so you should already have that ready to go. So in terms of stuff that you should already have done, title is really straightforward. That'll take you about a minute to do, so that should be completed. The question, we wrote that prior to starting the lab, so the question should already be done, and that's based on that purpose statement that you had. Your background research. Your background research should also already be done. I won't be around to check it today, but I will be checking it on Friday. So you get a grace if you don't have it done today. I will be checking it on, not Friday, but Thursday. Sorry. Background research. Your background research is 2 paragraphs. The first paragraph, you need to define osmosis. Then you need to tell me what's going to happen to a cell in either a hypertonic, hypotonic, or isotonic solution. So that's your first paragraph. Your second paragraph is you're selecting one of the 3 bulleted points. You're doing a little bit of research on it, and you're explaining to me how osmosis affects the absorption of water in roots of plants or the absorption of water in your digestive tract, your small intestine or your large intestine. So you're going to take one example and then you are going to do a little research on it. You need to cite 2 sources. My suggestion is cite the textbook and then cite your other source. Remember that you can use on Google Docs, there's an add-on called EasyBib that makes it very easy for you to cite in APA format. So your background research, again, should also be done. Your hypotheses need to be in the form of an if-then statement, and there should be 3 of them, and again, these should also already be done. In terms of materials, we walked through the materials list, so you should also have this completed. So your materials list should also already be done. Be sure to include both size and quantity for your materials list. Diagram of your setup. Your diagram of your setup is just basically a diagram of the beakers all laid out, the eggs in the beakers, the solutions that were there. So you're going to, I think I drew a simple sketch when we were first setting up the lab of what the setup was going to look like. Just make sure you label your setup. So your setup needs to be, move this over here. Your setup needs to be labeled. So make sure you label your setup. Okay, so this means with amounts of things, if we're talking about liquids and other things like that. So diagram of the setup, this may be something you need to work on. So I'm going to put a 0 there. Let's scroll down and move on. So procedure. The procedure is probably the easiest portion of your lab report at this point, considering the fact that it's pretty much written for you. The only things you need to alter is you need to change the 130 milliliters in the procedure to 150 milliliters. You also need to be sure that you include the day 1 procedure, which is the decalcification procedure. Even though you guys didn't technically do it and I did it for you, you do need to include it because a lab report somebody who reads your lab report needs to be able to replicate your experiment, and that involves decalcifying the eggs prior to putting them into the different solutions. So please make sure you include the day 1 procedure as well. So in terms of procedure, that should also be done for as well. Data table. This should pretty much be done. You might need to retouch it, retype it up, make it look pretty, but that should pretty much be done. In terms of a graph, you can either choose to do a bar or a line graph. If you do a bar graph, you'll be doing a bar graph on the average of the percent change in mass. So you would take your 3% changes of mass for your hypertonic solution, or 4 or 5, depending on how long we did it for. Add those all together, divide them by 5. Get your average percent change in mass. You're going to be creating 3 bars. For your line graph, if you do a line graph, remember that time will be on your x-axis, hint, hint, nudge, nudge, wink, wink, and you'll be having 3 lines. So be sure on your graph that you're going to need to include a key because you've got 3 different types of solutions going on. You can choose to use graph paper, so if you're not comfortable doing a graph on the computer or you're not sure how to do it, you may use graph paper. Any questions, comments, or concerns? Please email me about the graph, or I can help you out during tutorial sometime later on this week. So the graph might be something that you still need to work on. All right, your conclusion paragraphs. You're going to have 3 conclusion paragraphs, and that's in that packet that I gave you that has the lines on it, and it also has the rubric that I'm going to be using to grade attached. Your conclusion paragraphs. Be sure to use data. Okay? You're primarily going to be using data in the first paragraph. So for paragraph 1, take a look up here. Okay, paragraph number 1. What you are going to do is you are going to restate your hypothesis number 1. So essentially you're just going to copy and paste. You're going to tell me whether or not your hypothesis is either supported or refuted. Your hypothesis is not correct. Your hypothesis is not wrong. It's either supported or refuted. So make sure that if you did write right or wrong, or correct or incorrect, that you change that to either supported or refuted. Then you are going to go on and discuss your data. Make sure that when you do discuss your data, you're including units in there. You don't want to forget your units. So in this case, we're dealing with grams. And you're also going to talk about the movement of water. So for your hypothesis number 1, you might have stated that if your egg was placed in a hypertonic solution, then the mass of your egg will decrease in size. You'll go on and talk about your data, like the initial mass of the egg was 70 grams, and the final mass of the egg was 68 grams, and there was an overall 2% change in mass. This shows that water moved from an area of high concentration in the egg to an area of low concentration, the syrup, showing that syrup is in fact a hypertonic solution. So make sure you talk about each of these 3 things. Then you're going to go on and talk about your hypothesis number 2. Do the same thing, discuss the data, discuss the movement of water, and do the same thing for hypothesis number 3. So your first paragraph is going to be a pretty big paragraph where you're going to address all 3 hypotheses. In paragraph number 2, you are going to talk about any problems that you might have encountered with the procedure, any possible improvements you might make to the procedure, and then talk about further experiments. With regards to the further experiments, think about solutions that organisms would encounter in the real world. Maybe, I don't know, freshwater, saltwater, treated water. So think about that. And in paragraph number 3, You are tying in what you've learned about osmosis as a result of this lab, and you're tying it back to your second paragraph in your background research, so how it applies to real life situations. Again, this is all outlined in the rubric that I gave you. So be sure when you're writing your conclusion paragraphs to also use the rubric to help you. That's why it's there. Use the rubric. When you are, a couple of notes that I want you, couple of things that I want you to be aware of. Really one main thing. When you are talking about your eggs, okay, do not tell me that your egg grew. Your egg did not grow. Growing implies that there was cell division, which means that at the end of your 50 minutes, you're telling me, if your egg grew, that you ended up with 2 eggs. That probably did not happen. If it did happen, please let me know, because that's a miracle. But your egg did not grow. So that implies cell division. You can say that your egg increased in size, your egg swelled, your egg shrank, but your egg does not grow. It's not growing in this lab. That implies that there is cell division going on and that you ended up with 2 eggs at the end of your lab, which odds are probably did not happen. Now going on to due dates. Here's the due date schedule. So a draft of your full-blown lab report, typed up, printed out, ready to go, is due on Monday, 12/15. So that's next week, so next Monday. Okay? I want a full-blown lab report. Title, question, background information, hypotheses, everything typed up, ready to go, ready to be turned in to me on Monday. Okay? Because we are going to be self-grading those lab reports and self-assessing those lab reports on Monday. I need those ready to go. If they are not ready to go, or if you do not have your lab report ready to go on Monday, that will be a minus 10 points off your overall grade. So the highest grade that you could earn on the lab report will be 50 out of 60 versus the 60 out of 60. So you need to have that lab ready to go for me on Monday, because we're going to be using them in class to help self-assess. Once we self-assess on Monday, your final overall due date is Wednesday, 12/17. So on Monday, you'll find out whether or not you need to make any adjustments to your lab report, and I will give you that time in order to make those adjustments. So that final due date is Wednesday, 12/17. That is a drop dead date, which means that if your lab report is not turned in to me by Wednesday, 12/17, I will not be accepting it, and it will be a 0. This lab report is for a total of 60. Take a look over here, 60 summative points. Summative stuff counts for 80% of your grade. This lab report can be a very good grade booster. So if you're on the cusp of a grade, this will definitely help boost you up into the next grade bracket. If you don't do so well, it could bring you down into the lower grade bracket. So be aware of that. On Thursday after your test, I will take any questions about the lab report. So if you do have any questions from today, be sure to jot those down so that I can answer any questions that you have. I hope this helps, and best of luck.